Welcome to AP Biology. This next video is going to be about the stages of mitosis. So, the easiest way to remember the stages of mitosis is pea mat. So, I always tell the kids, think about a mat you would buy for your dog. If you lived in a high-rise apartment in New York and you didn't want to get up every morning and walk your dog at 6 a.m., the dog could just take a quick little pee on this little mat here and you can clean it out periodically. And then you wouldn't have to take the dog out quite so early. So, P-M-A-T. Prophase, metaphase, anaphase, telophase. So, this process we're going to talk about is a continuous process. It doesn't really happen in stages. It's not like we go to prophase, then we go to metaphase, blah, blah, blah. It's like a continuous process that takes about an hour. And it kind of goes through the process of moving the chromosomes around and then separating the sister chromatids and forming, tearing apart nuclei and forming new nuclei. But it's a continuous process. So what happens during prophase? Well, prophase is the beginning of this process, and there's a few key things that are going to happen. First, your chromosomes are going to condense, so the chromosomes are going to become visible because they're going to pack themselves real tightly into chromosomes, I guess. So you have chromatin that is kind of a stringy form, and it's going to coil around histones, and then it's going to form a supercoil, and then it's going to form a super duper coil, and then it will form into chromosomes. The centrioles are going to move to the opposite sides of the cell. As the spindle fibers grow, they're going to push the centrioles to the opposite sides of the cell, and then the centrioles are going to anchor to the cell membrane on the opposite sides of the cell. The spindle fibers are preparing for grabbing onto chromosomes and moving them around, and the nucleolus is going to dissolve, and then the the nuclear membrane is then going to dissolve following that, and that will start the beginning of kind of a pro-metaphase stage. So this is the packaging of DNA. So here we have the nice loose DNA molecule. It wraps around balls of histones, and then it's going to coil again. So we call this coiling, super coiling, and then super duper coiling, and then we form our chromosome. So this chromosome is going to be made up of two sister chromatids. Okay, each one of these is an identical copy of this. Okay, so these are the sister chromatids. This is the original chromosome. So you got two identical copies. So when the cell divides, one copy of each chromosome goes to each new cell. This is what it looks like under an electron microscope. Scanning electron microscope, you can see the centromeres of the chromosomes, and then this would be one copy, and then this would be the other copy. All right, so as we transition to metaphase, we're going to go through something they call pro-metaphase, which is really not part of PMAT, but basically what happens is the nuclear membrane dissolves, and then the spindle fibers are going to grab onto the chromosomes, and they are going to move those chromosomes and line them up in the middle of the cell. Basically, we're just preparing for an organized way of dividing up the chromosomes. Once we reach metaphase, where the chromosomes are all lined up in the middle, okay, there will be some checks that go on to make sure that everything's lined up in the middle properly and that we are ready to divide. Once we are ready to divide, then we will make our way into anaphase. In anaphase, there is a protein that holds the centromere together, and it's called cohesin. And cohesin keeps this centromere in one piece, and at the appropriate time, when the signal has gone out, there's an enzyme called separase. Kind of easy to remember, separase, because it separates the ends or separates the sister chromatids. It's going to go and it's going to dissolve the cohesin and allow these sister chromatids to be pulled to the opposite ends of the cell. Basically, the spindle fiber just shortens and it gets pulled to the opposite ends of the cell. It's important that the centrioles anchor themselves to the cell membrane because they're not very big and they're going to be pulling, in some cases, lots and lots of chromosomes to one side of the cell or the other. In humans, they pull 23 or 46 to each side, but in other organisms, there can be even way more than that. And then we have telophase. In telophase, the DNA will start to unravel back into chromatin, but first we need to form a new cell or nuclear membrane around each sets of DNA. So that's going to happen first. Nuclear membrane will form, and then the chromosomes will start to break back down into chromatin so that they can be read and utilized again. And then following that, we're going to split up. So basically, mitosis splits up the nucleus into two separate nuclei. And then following mitosis is a process called cytokinesis, where we actually divide the remainder of the cytoplasm. So when we say mitosis, that's only division of the nucleus. Mitotic cell division is the division of the whole cell. So cytokinesis here, we are dividing up the cell. Basically what happens is there are actin and myosin filaments right here that are going to contract and constrict, and they form a cleavage furrow. And that cleavage furrow forms all the way around, kind of like if you take one of those long balloons and you start twisting it. There will be a cleavage that forms, and then it gets real skinny in the middle. And then once it gets real skinny, then the cell, this side of the cell membrane will combine with this side of the cell membrane, and the same on the other cell, and you'll get two new cells that will plop apart from one another. It's a little bit different process in plants. In plants, instead of pinching together, because they have a rigid cell wall, so they can't pinch together very well. So instead of pinching together, in plants, a new cell wall will start to form, and then that new cell wall will fuse with the existing cell wall, and then this thing will become actually two cell walls, and so one will fuse with this side and one will fuse with this side, and then you have two separate cells.